Greetings, my chief.
Step into MedNemo the lab, where mnemonics make med learning vivid. We're culturing clever memory hacks to help you isolate key topics. From vaccinating against forgetfulness to transfusing mnemonic magic, we've got the remedy for med school stress.
It's Sarah here, and today we'll be talking about the different receptors types in pharmacology. A receptor is a specialized macromolecule, usually a protein, located on the surface or inside a cell that interacts with a drug, ligand, to produce a biological response. In simple terms, a receptor is like a lock and the drug is the key that fits to open it. Types of receptors. There are 4 types of receptors: ligand-gated ion channels, G-protein coupled receptors, enzyme-linked receptors, and intracellular or nuclear receptors. Ligand-gated ion channel receptors, also known as ionotropic receptors. These are membrane-bound receptors that directly control ion channels. When a ligand, drug, or neurotransmitter binds, the channel opens and ions such as sodium, potassium, chloride, and calcium ion flow into the cell. This causes a change in cell membrane potential, leading to a rapid response. The response time for this type of receptor is very fast, happening within milliseconds. Examples of ligand-gated ion channels include nicotinic acetylcholine receptors, N-sub-M and N-sub-N, GABA-I receptors, glutamate or NMDA receptors, and 5-HT3 receptors. The ligand for nicotinic receptors is acetylcholine or nicotine. When they bind, there is sodium ion influx, leading to skeletal muscle contraction. The ligand for GABA-A receptor include GABA and a class of drugs called benzodiazepines. When they bind, there is chloride ion influx, leading to hyperpolarization, aka inhibition. The ligand for glutamate receptor is glutamate. When it binds, there is calcium ion influx, leading to excitation. For 5-hydroxytryptamine type 3 receptor, the ligand is serotonin. When it binds, there is sodium ion influx and potassium ion efflux, causing depolarization. This depolarization stimulates the vomiting center. G-protein coupled receptors, also called metabotropic receptors. They are 7 transmembrane proteins. Binding of ligand activates a G-protein, which alters an enzyme, like adenylyl cyclase or phospholipase C, causing them to generate second messengers like cyclic AMP, IP3 and DAG, which then produces a cellular effect. Response time is in seconds. G-protein coupled receptors have 3 subtypes: GS, GI and GQ. GS receptors stimulate the enzyme adenylyl cyclase, thereby increasing the level of cyclic AMP, a second messenger. It has a stimulatory effect on organs such as the heart and the bronchi. GI receptors inhibit the enzyme adenylyl cyclase, decreasing the level of cyclic AMP. It has an inhibitory effect on the heart and CNS. GQ receptors activate phospholipase C, which cleaves PIP2 into inositol 3-phosphate and diacylglycerol. IDP3 then binds to receptors on the endoplasmic reticulum, causing the release of stored calcium. This then leads to contraction and secretion. Examples of GS receptors include beta 1, 2, and 3 receptors. Their stimulation cause increased heart rate and bronchodilation. Examples of GI receptors include M2, alpha-2, and dopamine type 2 receptors. Their stimulation decrease heart rate and norepinephrine release. Examples of GQ receptors include alpha-1, M1, and M3 receptors. Their stimulation cause vasoconstriction and decrease secretion. Enzyme-linked receptors, also known as kinase-linked receptors. These receptors are single transmembrane proteins with an extracellular ligand binding domain and an intracellular enzyme domain, often tyrosine kinase. Ligand binding to this receptor leads to receptor dimerization. Autophosphorylation. Activation of enzyme cascade, which finally leads to altered gene expression and metabolism. Response time ranges from a few minutes to hours. Examples include receptor tyrosine kinase, cytokine receptors, JAK-STAT, and guanylyl cyclase receptors. The ligand for receptor tyrosine kinase include insulin, epidermal growth factor, and platelet-derived growth factor. Binding of the ligand activates RAS and PI3K, causing growth and metabolism. The ligand for cytokine receptors include growth hormone and interleukins. The JAK STAT mechanism begins when a cytokine activates a cell surface receptor, which in turn activates Janus kinases, JAKs. These active JAKs then phosphorylate STAT proteins, allowing them to form dimers. Enter the nucleus and regulate gene transcription. The ligand for guanylyl cyclase include atrial natriuretic peptide and nitric oxide. Its binding causes increase in cyclic GMP, leading to vasodilation. Intracellular or nuclear receptors. They are located inside the cytoplasm or nucleus. The drug, usually lipid-soluble, diffuses through the membrane and binds to the receptor. The complex then binds to DNA and alters gene transcription, leading to the formation of new proteins. Response time is between hours to days. Intracellular receptors include steroid hormone receptors, which are situated in the cytoplasm. In the nucleus, we have thyroid hormone receptor, vitamin D receptor, retinoic acid receptor, and peroxisome proliferator activator receptor, PPAR. Steroid hormone receptors essentially regulate reproduction and metabolism. Thyroid hormone receptor regulates metabolism and growth. Vitamin D receptor, which regulates calcium and phosphate homeostasis. Retinoic acid receptor, which regulates cell differentiation, and finally the PPAR receptor, which regulates lipid and glucose metabolism. Check out my next video for mnemonics. Until next time, stay safe, stay happy. This is Mnemonic-based medicine.